Teacher, can you say a few words so that we can know? Unmute your microphone and say a few words. Yes, hi. I'm a teacher. Hi, teacher. Can you say a few words? Hello,
Sunita.
Thanks, you're audible. Thanks. All the participants are here. Also, we have Bhaskar. Can you change the guy from Nigeria? What are the scientists from Nigeria who wanted to join us? So I have to be here for video. I welcome you once again to the lecture session. The first lecture will be from Professor D.K. Higgins. He is an honorary fellow at CSIRO Agriculture and Food in Australia, Canberra, Australia. He is also a project member of the Carby research collaboration. We collaborative project. We have a long association with TJ. The association started way back in 2004. I think before 2004, February 1999, when Professor Vinod Kumar Sharma introduced the contributor to do his postdoctoral research on chickpea, not chickpea, alpha-virus chickpea. Since then, we have collaborated with TJ. All of us, most of our scientists from our laboratory visited TJ. I myself visited TJ. I was there in his lab recently in 2018 and 19 to do some biosafety related work on BT-chickpeas. So TJ, to introduce TJ, he is an outstanding example of distinguished contribution to basic science heading to a range of application in agriculture which are of national and global significance. He initiated, work in BT copy way back in 2000, and he has successfully generated copy lines which are commercialized in 2019 in Nigeria and several other states in Africa. So that's a huge contribution. He has generated BT protected to record barbican, and it's a big contribution to the small farmers there in Nigeria. He is widely recognized internationally for his contribution to outstanding work in the area of plant biology, molecular genetics, or storage protein found in the developing seeds, the regulation and expression of the encoding genes. These studies combined with his research on gene technology provided the basis of generating transgenic crops and pastures in detail to provide critical novel features. These include plants with either improved nutritional quality, resistant to pests such as potholders, and also storage interests like ingredient. I welcome DJ to speak and deliver a lecture. The topic, I think teacher you will be telling you the topic. I think I don't have the topic with me right now. So over to teacher. Teacher, please continue. Not only for the Hello teacher, you have a
Hello, sir. Please say something.
DJ?
Okay.
Yeah, you can start your lecture, please. Yeah, it's visible, teacher. Just put it on slideshow mode now.
Hello, can you hear me?
Yeah. Yes. Yes.
Can you see the picture?
Yes.
Okay, great. Thank you for the invitation. I greatly appreciate having a chance to talk to friends at Assam Agricultural University. It's a big honor for me. Today I'm going to talk to you about a gene technology example that I think suits smallholder farmers. The example I'm going to use are smallholder farmers in West Africa, but they apply equally to smallholder farmers elsewhere in the world, including in India. And what I'm really talking about is food security. And while food security is a complicated issue involving poverty, governments, government institutions, it also relies on the production of food, first of all. And food production relies on, amongst other things, good crop genetics and good agricultural practices. I'm going to be talking mostly about crop improvement through genetics and plant breeding, but good agricultural practices, good agronomy is equally important, if not more important in many cases. From a long time ago, before Mendel, farmers mostly, were improving their crops, back probably 10,000 years. I'm just going to move to when we started actively using crossbreeding about 120 to 130 years ago. Hybrid breeding came into use in the 1920s, just over 100 years ago. Tissue culture has been used to improve crops probably about 60 or 70 years ago, 1960s. And more recently, gene technology and other smart breeding techniques, including genomics, have been introduced in the last 40 or 50 years or so. So some of these techniques are very recent. Plant breeders have access to many tools. Crossbreeding and selection, interspecific crosses, and intergeneric crosses would be familiar to most of you. But mutagenesis, hybrids, and double haploids are also available a bit more recently. Molecular markers, better statistics, and more recently genetic engineering and gene editing. So you can see that plant breeders have access to a very wide range of tools, depending on the problem that they're trying to solve. I'm going to focus on just one food legume for today, that is cowpeas, which I think, which Sumita has just mentioned. This is an indigenous crop in Africa. It was first domesticated in Africa about 4,000 years ago. There are many Calpe breeders in West Africa, and these are three that have been vital parts of the project that I'm going to talk about to you today. This is JB Tinigwe in Burkina Faso. This is IDK Atoptle in Ghana, and this is Mohammed Ishiaku in Nigeria. These are the national breeders for each of those three countries, and that there have been, as I said, a vital part of the work that I do. They're my long-term collaborators. I've been working with them for about 20 years or so. Just a few facts about Calpe. It's a food legume, as I sort of indicated, or often called a pulse. It came from Africa. It has high protein grain, like chickpeas and other food legumes, 25 to 32% protein. It has a low glycemic index starch, good quality starch. Global production of Calpe is quite a lot less than chickpea, but it's still 10 million tons annually. So it's still quite a significant crop. It fixes nitrogen and has high feed value. Even the stubble that's left over after harvest is valuable for feeding livestock. Pests and diseases are major problems, however. And one example is shown there on the right-hand side. This is a moth called Maruca vitrata, or the pod borer. It's a very significant pest, and in bad years can decimate probably 80 to 90% of the crop, but on average probably reduces yields by about 30%. Now, Calpe is a crop of smallholder farmers. These are farmers who have less than 1 hectare usually. And it's usually intercropped with tall crops like sorghum or maize, as is shown. Sorghum is shown here. So you can see the technology is low tech. It's very different to many other parts of the world. This is an example of a farmer, a smallholder farmer. He's getting to be a slightly bigger holder farmer. His main crop that he grows is Calpe. He says it's his livelihood, and the challenges that he faces is this Calpe pod borer, Susan Waki, which he controls by spraying up to 7 times in some seasons. But this has become more common over the last 10 years or so, as chemical insecticides have become cheaper. The farmers have been spraying sometimes up to 10 or 12 times a season to control the pod borer. As I said, it's a serious pest. This is the sort of damage that it does to the pods. The pods, any pods that do survive, look like this, but not very many do survive. The moth lays her eggs on the young flower buds, and the larvae begin their growth inside the flowers. They develop fully on the pods and especially on the seed. And as I said, yield losses often exceed 50%. Sometimes much more, sometimes a bit less. Insecticide use is very informal in West Africa and can be quite dangerous because large samples of insecticides are subsampled into soft drink containers and often have no label on them at all other than the fact that they're an insecticide. So it's very dangerous. The options that the farmers have is not to control the insects and to rely on the seed's natural resistance, of which there is not very much. The whole collection, 16,000 accessions, have been tested for natural resistance, and basically there is none in the international collection at IITA. And yield is about 350 kilos per hectare if they don't spray. If they use insecticides, they can greatly increase their yield, probably up to nearly 2 tons per hectare. But farmers are not very keen to use chemical insecticides, and many farmers, and certainly the plant breeders, would like the option to use biotechnology to introduce inbuilt resistance, as has been introduced into other crops like soybean, another legume, for example. We don't know what the yield increase would be with the biotechnology, but towards the end of this talk, I'll give you an indication of what the yield might be if we could at least control this insect pest shown again there on the right-hand side. The technical aspects of Calpe gene technology are very straightforward. It's been practiced for about nearly 40 years now. A gene transfer system had to be developed for Calpe. Genes had to be reconstructed into a plant format so that the genes would work in a plant rather than in a microbe, which is where some of the genes came from. The genes can then be transferred into Calpe if you have a gene transfer system, as is indicated in number 1 there. And once the genes have been transferred into Calpe, their molecular analysis of the transgenic lines is conducted over a period of years. And field trials eventually, if they survive all of these tests, field trials to select a unique event or the best line is the next thing to be done in the field in West Africa. And then, as soon as a unique line has been selected, the plant breeders, the people that I showed you at the beginning, will then use that material as a parent in their breeding program into their best varieties. And they've been, of course, they are producing new varieties every few years on a regular basis. The transformation gene transfer system that we used for Calpe was based on Agrobacterium tumefaciens. We used a DNA plasmid vector for gene delivery. And we used kanamycin selection, NPT2, neomycin phosphotransferase 2, to select out the lines that have taken up the new DNA. And we use explants, starting material from embryo axes of imbibed mature seed. You can see the great variety of cowpeas on the right-hand side there. They're not all black-eyed peas. Some are quite black, totally black. Some are brown, some are red. All kinds of colors. Our Calpe transformation system, as I said, starts with mature seed. They're imbibed overnight and split apart. The seed coat is removed and the seeds are split in half and incubated with Agrobacterium. They're then tissue cultured for several months, presence of sugars and other nutrients. Eventually, shoots start to develop on a shoot induction medium and then are transferred onto new media where roots can be induced to develop. Those rooted plants are then transferred to soil where they are acclimatized to lower humidity and eventually transferred into a greenhouse where they flower and produce pods, as is shown there on the right-hand side. The first gene that we have used is a Cry1AB. It's a BT gene, crystal protein 1AB, for protection against this pod borer. There are 2 genes in this binary DNA plasmid. One is the NPT gene on the left-hand side, upper left-hand side there, and the Cry1AB gene on the upper right-hand side. But those 2 genes are then transferred into the plant chromosomes. We screen our transgenic lines in the lab and in the greenhouse or glasshouse. We look for high levels of expression, especially in the flowers, because that's what the larvae of the pod borer attack first. We look for single copies of the transgene. That's so that, because this is a single dominant gene, we want it to be at a single locus, which makes the plant breeding downstream much easier. We don't want any other DNA other than the transgenes in there. Of course, we select lines that are homozygous and have a good phenotype, vigorous phenotype. We do laboratory bioassays here in Australia with the Maruca larvae before we send anything to Africa. We send are the seed of selected lines to Nigeria, Burkina Faso, and Ghana, as you can see there on that map of West Africa. The crops are grown in northern Nigeria, southwestern Burkina Faso, and northern Ghana. The typical layout of a field trial is typical of most trials that plant breeders conduct. Here I'm showing you 6 different transgenic lines across the top there, as well as the control line, IT86, in red. This is a replicated trial, very typical of the sorts of trials that are done in the field at any time. These trials are subject to biosafety laws in each of the countries. The trials are protected from external influences. People are not allowed into these trial sites unless they have special permission. One of the very important things that the plant breeders insist on is that there is no yield drag associated with the transgene in the field. This is, of course, a common problem is that when plant breeders make new lines, sometimes for disease resistance or even insect resistance, there is yield drag associated with that trait. So one of the first things that we did was to test some of our best lines in the absence of insects to make sure that the transgenic lines shown here, 709 and 252, are no different in yield to the parent line, IT86. I'm showing you their yield, seed yield in grams per plant, which is pretty typical, 25 to 30 grams of seed per plant. And you see there is no difference between the transgenic lines and the parent line. Now, this is in the absence of insects. So this convinced the plant breeders that there is no yield drag associated with the transgene in the field. And this is a typical field trial showing you the 2 lines, the non-transgenic line on the right-hand side and the transgenic line on the left-hand side. You can see very few pods on the non-transgenic line, I hope, and lots of pods on the transgenic line. This is now in the presence of insects. These fields are infested artificially with high levels of insects. Larvae are put on the plants every second or third day for a period of 15 days or so. So we really test the transgene as to whether it's having any effect. And as you can see there, it does have an effect. And this is some quantitative data showing again now plants grown, 2 of the lines out of, we tested about 32 lines in Africa out of the many lines that we made. Here are 2 of those lines again, 709 and 252, compared to the parent line. This is now seed yield again on the y-axis in grams per plant in the presence of insects. You can see that IT86 yields about 4 grams per plant, whereas 709 and 252 yield between 14 and 18 grams per plant in this particular case in this trial. That's in Nigeria. In Burkina Faso, the insect infestation was higher that year, and you can see that there was basically no yield of the non-transgenic, and the 2 transgenic lines produced between 20 and 25 grams per plant. So this Cry1AB gene was giving very good protection against the pod borer. And this gives you an indication of the number of seed that you can get from a single plant in a field trial for 709 and 252D compared to the IT86, again, shown on the left-hand side there. This is number of seed per plant. Out of those lines, plant breeders, of course, make their final selection as to which line they would prefer to use as a parent. and they have chosen 709A as the parent. This is Mohamed Ishiaku looking at 709 in the field and making a decision that that's what he will use as a parent in his breeding program with his best material. The breeders have now made varieties in all 3 countries. I'm just showing you some data here from Nigeria. This is Calpe pods on transgenic and non-transgenic varieties now. This is a variety, not the line that I sent to them in the beginning. This is a backcross 4 line, a null segregant on the left-hand side, where the plants have produced very few pods, but the plants have remained green because there is no transport of material from leaves to the pods. Whereas on the right-hand side, the transgenic variety now is producing lots of pods. As you can see there, quantitatively, that's what this looks like here. The transgenic line produces about 20 pods per every plant. There's no pod damage, and the seed weight is about 30 grams per plant, whereas the null segregant variety, without the trans gene, but otherwise the same, null segregant, produces only 3.5 pods per plant, and over 95% of the pods are damaged, and they produce only 5 grams per plant. So the farmers are given opportunities to come and look at these trials in the trial site so that they can make up their mind as to whether the plants look normal, which they do, of course, but they need seeing is believing as far as the farmers are concerned, of course. So there are many information days for them to come and look at the plants. And in fact, the farmers get involved in the final selection of the lines that are going to be made into varieties. About 20 farmers participate in the plant breeding process, which is very important because they need to be convinced and to accept the lines, of course, that the breeder makes. So last year, the BT Calpe was launched for sale to farmers in Nigeria. This is certified seed being given to 4 different small seed companies that have been licensed to sell the seed. I would like to, first, before I get on, finish up talking about this, I'd like to thank the many collaborators. I mentioned these 3 plant breeders who have been absolutely vital parts of the project in Nigeria, Burkina Faso, and in Ghana, as well as entomologist colleague Larry Murdoch, particularly at Purdue University in the US, who's a vital collaborator in the early stages of this project, and of course my CSRO, agriculture and food colleagues here in Canberra. But very important in all of this, of course, is funding. The funding for the work has come largely from USAID and from the Rockefeller Foundation and CSRO, and I acknowledge them with gratitude for their funding of the work. I also acknowledge the African Agricultural Technology Foundation and Monsanto for access to the BT gene, royalty-free. There is no royalty attached to the use of this gene. I'd like to give special thanks to my colleagues here in Canberra, Lisa, Stephanie, and Andy, from left to right there, who've been vital in the tissue culture and molecular analysis of these plants in Canberra before they were sent to West Africa. This is just a picture of farmers looking at the BT Calpe, selecting the best varieties for release before they were released. So they were participating in what's called farmer participation in plant breeding. The BT Calpe was approved for release by the Federal Republic of Nigeria, the National Biosafety Management Agency, NBMA. So this certificate of permit allowed us to then take it to the next stage to start producing breeder seed and foundation seed. So to conclude, all of the biosafety studies associated with getting a crop like this released to farmers are now completed for this BT gene. It was released to the farmers in 2021, that last year, as I said, and we have had very positive responses from the farmers who had access to seed. We did not have a lot of seed last year to give to the farmers, but the seed is being bulked up. We will eventually need a lot of seed for the farmers, but we are doing this one step at a time. There's still issues, of course, to be resolved. Community acceptance, even though the farmers are consumers, of course, as well as producers, we still have to be assured that the community in general will accept this. And of course, we don't know this until the seed is widely available. So far, community acceptance is very high, but this is still very early stages. There's, of course, issues still to be resolved include varietal approvals in Burkina Faso and Ghana for dissemination to farmers. So that's still a challenge that we have yet to complete, although we are getting very close. Our major activity in the lab here is transforming with a second gene to reduce the chance of pod borers developing resistance. Insect resistance management is very important, and that's what we have been doing, is adding a second gene, and we're well advanced with that now. The pod borer resistant cowpea is out there. However, there is still, as you would well know, there is still a lot of debate around GM. And the facts often don't matter all that much. This is an example here where the person on the left is saying that her answer was correct. Jenny's answer was correct, but Kevin shouted his incorrect answer over yours, so he gets the point. And there are a lot of incorrect and misinformation answers about gene technology out there. And it is a major challenge to deal with this, informing the public. It might not be a good idea to point this out in general, but this paper in Nature a couple of years ago said that extreme opponents of genetically modified foods know the least, but think they know the most about GM, and frequently make all kinds of claims that have no real information at all. In fact, they're usually very often quite wrong. So that's one of the other issues. It's all very well for people like myself doing this sort of thing technically in the lab, but there are many other issues to be considered talking about it in the lab. I'd like to finish there and perhaps ask if anybody would like to ask a question. Does anybody have a question or comment? I think people are there. Are they?
Thank you, teacher.
You're welcome.
Presentation. Yeah. So seminar is open for discussion. So if you have any specific question, you can ask DJ, and I'm sure he will be happy to answer. Anybody? Nothing. Yes, go ahead. You have something? Yes. Yes. Yeah, take this mic and say.
So we are taking the BTGs,
Yeah.
everything about the VIP disease.
DJ.
I didn't hear the question.
Okay, you come here and say. Okay. What's the book?
So we are taking the BTGs. Is there any things about the VIP3 genes? What we have taken?
DJ is asking about BIP3. He's asking about BIP3.
Asking about what?
BIP gene. BIP. VIP gene.
Sorry, I didn't hear.
Can you just type? Just type.
Am I audible now, teacher? Hello, teacher. Am I audible to you now?
No, I'm sorry, I can't hear the question. Can you put the question in the chat box?
Yeah, I'm doing that.
Oh yes, I see now. Yep.
He goes, teacher, chat and लिखिए सब. Hi, teacher. You don't
I'm
need,
writing an answer.
no, you don't need to write. You can say because you are audible to us. Yeah. He will sound to enjoy that.
So what I have written is that we are looking at a VIP3B gene, but are concentrating on a Cry2AB gene first.
Okay, thank you.
We have a lot of data on Cry2AB as being very effective against Maruca, but we have less data on the VIP3B gene. We have some data, but not as good as the 2AB gene.
So I think participants, you know what is VIP3 gene. It's a vegetative insecticidal protein, which is again from reference lineages and equally effective against lepidopteran pests. So what TK says that he is concentrating on Cry2AB gene, which is a principally protein secreted by the bacillus, the same bacteria. But eventually, possibly he will also go for pyrimidine, where you will have three different genes, like BT content, which is available commercially, has three different genes, Cry1AC, Cry2AB, and BIP3. So possibly the Carby he has been planning to do so as well. Okay. More questions? Yes. Okay. So participants, you have any questions? DJ, you have to ask them questions probably. That's what it is. Usually students they don't interact. So we have to make them, talk a little bit in this lecture session. There's participants from Harvard University. Anybody wants to know more about DBT-Carby? You have to know that this is the first, food crop that has been deregulated. First, the new food crop, possibly. It's very great work, and I'm sure the farmers in Nigeria will be benefited by this work that TJ has done. So it's a huge contribution from TJ and his group to the people in Africa, and eventually continue to be integrated in other countries as well. Although in India, coffee is not a popular crop, but maybe similar kind of technology may be used for other legumes like we are doing for chickpea and PGP, another popular legumes that are grown in India. Okay. TJ, you have something more to say? Because students are not going to ask any question to this. All right, yes. Yes. You want to come here and come. Come. TJ, am I audible to you?
Sir, I would like to ask you whether agarobacterium bacterium efficiency is more effective or Ispercasmani is more effective in terms of your research.
Subita, can you type that question into the chat box?
Can you hear me? Am I audible to you, TJ? No. Okay, I'll type it. Possibly he's typing.
I have sent an answer back saying we have no experience with CRISPR-Cas, we only with agrobacterium. So that's what we are using now. Our work is largely GM, not gene editing. Although I'm very pleased to see that India is now accepting SDN1 and SDN2 as being non-GM, which could be very good for the future in India. But for the topics for us, gene editing is not necessarily all that useful. We do need to add in genes like the Cry1AB and 2AB. We don't have good candidate genes to edit in Calpe with CRISPR, which is why we are using agrobacterium and whole gene transfer.
Have you understood what he told? Often, when you want to improve crop plants, so you want to add some genes, or you want to do silencing of a gene, anything that you want to, or modify a gene, you need to, we have options. Earlier we had few options, like we had a gene, we are taking it from the bacteria and we are trying to put it into the compound, right? But now we have CRISPR technology where you can do some single nucleotide or you can cut a gene as a gene. But in some of the cases, especially for the insects, the insects we are working with, TJ talked about controllers, we don't have any candidate genes available. The only option that is available with us and which is most effective is your PTG. PTG has given almost, in our case, in TJ's case also, it has given us almost 100% resistance in the field condition. So something, a very good candidate gene when you have, the only option is you know put it through your bacteria. Because if you use the gene, you can put the gene using the CRISPR system also, but the term that is used for that is called as SDL3, where you are having an additional gene added to the genome. Okay? When you have a foreign DNA inside the crop plant, then it will be considered again as a GM crop. Okay? But in this case, in most of the cases, I must say, in case of the insects, we don't have a candidate gene to mutate, to edit, or do something. Only options that we have is a foreign DNA material, which gives us an effective control against the pest. I would like to request online participants, if I am audible, also to write their questions in the chat box. Online participants, any more questions? Anybody? Yes? Women empowerment. We talked about that in the morning, and only 2 boys came up and asked questions. Please, girls. You just tell me the question, I'll convey it to you. Yes, again what? Yes. Come in. It's not audible. You're talking about non-target.
Yes.
So yes, I've got that second question, that new question. What about non-target insects? This is work that has been done by colleagues in Africa, entomologists who have looked at non-target insects and feel that, and they find that there is no effect of the cry protein on non-target insects as far as they can measure, whereas chemical insecticides affect everything, beneficial insects as well as the pod borer. So the Bt protein is a much safer option as far as not only the farmers are concerned, but also the beneficial non-target insects, which do survive and can help control any maruca that survives. So yes, this is a big question that the regulators posed for us at the very beginning, and a question which, as part of the biosafety studies, we had to provide answers and data. This was work that we did not do this work. This was work that was done by entomology colleagues in West Africa.
Any more questions? If you don't have any questions, then I'd like to thank TJ for his active participation. I don't know whether I'm audible to him or not. Thank you, TJ. Yes, you have a question? Yes, again about it? Yes, okay. Okay.
What about the Calpe incidents? And
What about?
Calpe
What about coffee behaviors?
that is also on the answer Facebook. In transit, essentially what the reason for the part.
Yes, I got that question about does the gene have any effect on Calpe aphids? No, this gene, the CRA genes, do not affect aphids. But the plant breeders assure me in West Africa that they have some good accessions that they can use for aphid resistance in Calpe using molecular markers in their conventional breeding program.
So you have to understand one more thing is all the biotechnological approaches or tools that we are using to generate improved crop is basically going to complement the breeding technology. We are not creating something different. We are trying to help breeders wherever the breeding techniques are not useful, such as in the case of insects. We don't have any genes, or we don't have any genes in the gene pool to make crosses. We don't have any resistant blind against insects, including the aphid that we talked about. In that case, we may have to go to the gene technology, tools. So for aphids, there are genes available which are called as lectin genes, so that are effective against, and some of the researchers are doing that. But if you know a candidate gene which can be used for breeding, or you have a gene in the gene pool, or a germplasm that is resistant to aphid, you can definitely cross it with a BT plant. So that in that case, the crop plant will have resistance against aphid as well as against the pod borer. So you can make those strategies. Okay. Any more questions? Again, I'm expecting at least one question from a girl. No. Richa? Maybe? No? It should not be one-way traffic. Those who are participating, it should not be one-way traffic. You have to speak, you have to participate, you have to make it much, much more lively for the speakers. Okay? Do a little bit of homework before you come to the lecture sessions, and then prepare yourself a little bit and make sure that you are asking questions. Okay? Because getting an opportunity to speak to TJ, TJ has received Australian order, which is equivalent to Padma Shri, a Padma Bhushan that is given here in India. He is a very renowned scientist. He has been working since 1975, before I was born. So he has vast experience, and if you spend some time talking to him, you will know that he has immense knowledge not only in gene technology but also in plant molecular biology, because almost 20 years he worked in plant molecular biology. You have his CV with you. We spent a huge amount of time in the University of California working on seed storage protein. So anything that you can ask anything about, if you want to know about molecular biology, any questions you can ask him. The interaction with this kind, such an eminent person, I think it's a lifetime achievement, and it's a lifetime achievement for me, really, because meeting him in person and always chatting with him is kind of a very good experience for me, and I have learned a lot from you. And this is the platform that I am giving it to you, and we, the organizing committee, giving it to you, so that you can interact with people, eminent scientists like you. Okay, take this opportunity. Okay. Okay, then we are closing. Thank you all for participation. Thank you, DJ.
We have a girl with a question.
Okay, we have I think thank you, DJ. Now you have a question. Yeah, most welcome.
You mentioned about how we are generally intertrophic crops, right? It's true.
How we are intercrops. Who? All crops. Let's come and type it. How we are crossed it.
Why Calpe is intercrop with all plants?
Intercrop with. Okay, thank you. Is it okay? Am I having the right question? Okay. So this is a question from agronomy, possibly. Yes.
Calpe is intercropped with tall plants because it's a climber. So the smallholder farmers do a lot of crop rotation this way, and the cowpeas fixed nitrogen, and the sorghum or millets or maize, those cereals, use the nitrogen that's fixed by the cowpea. So it's intercropped with tall plants just in order to get to the light. Thank you to everyone, and especially to Sumita, who seems to have given most of my lecture. Thank you very much. I greatly appreciate it. I might have her do this again in future.
So I'm audible to you, and you say you're not. You say I'm not audible to you, but actually I'm audible to you. Right? He's pretending again. I was in his lab in 2018, 2019, and whenever I say something, he will come to me and say, Sumita, I'm deaf. You have to speak louder. And he always did that, but I know he knows what I'm trying to say. He always pretends to be deaf. Okay? So anyways, thank you all. I think what we have is possibly some lab works for this. So DT, you can log out. You may log out. Please? He's finding something. And he loves children. He loves interacting with children. We always, try to stay connected with the young students. And although in CSIRO we don't have, it's basically an organization to do research only. There is no teaching facility there. But whenever he has time, he goes to Australian National University, which is just next door, almost 2 minutes walk. and he goes there and interacts with the student. Whenever any student visits his lab, he makes sure that he spends most of the time talking to them. The idea is basically to know that, to know what they are thinking, what kind of innovative thoughts they have, whether they are understanding the science in a better way. So he has always encouraged us to do science in a much, much better way. You know, and probably you have seen me, I'm just a lab rat, I stay in the lab so much, and that actually came from him. We say it always, you do good work and people will know you for your good work. Okay? He's typing still? Maybe he's doing some other ways. Anyways, before going, I just wanted to tell all the students, all the participants who are here, we have a bus. Bus will pick you up from the Dhanashri hostel and Barak hostel. Go for lunch. Don't roam around. Come back straight away for the lecture session because we want to start exactly on time. We don't want to delay it, especially the practical works. I don't know how long it will take, though it will be a kind of a method demonstration type. But sometimes you may have to be here till 5:30. Okay, and don't run away. Also, you have to complete this course. You have to be you have to participate in each and every lecture as well as practical works to get the certificate. We are going to take your attendance. Okay, before conducting the practical, we will take attendance as well. Okay, make sure that everyone is. We have arranged a lunch in the Dhanashri or Barak, one of the hostels. So take the bus and go there, have your lunch and come back. Okay? Come back at least 15 minutes ahead. Okay? So we are going to start our laboratory work on lab work from 2. Okay. Thank you all for today's morning session. Okay? Meet you in the afternoon. Wait, can you sit down, please? Just confirm one thing and what's to get up here? Well, thank you, teacher.
Thank you.
Bye.
Bye-bye. Thank you all for your attention. Very much appreciated.
Thank you so much.
And good luck with the rest of the symposium.
नमस्कार कोई है बास्केट अंदर हाँ बास्केट अंदर
1 बजे तो अभी ये लोग क्या करेंगे और वहाँ पे कैंटीन में नहीं Dhanashri में so the bus will come at 1. So what you can do is, we have a phytopark facility as well as a greenhouse facility. If it's not ready there, go there and visit and see the facility. Okay? All the participants. One of will take you to that.
So we go to the phytopark facility first and then
You are from our university? University students, you are not. Don't become less about this. Absolutely. You may not be able to become good. You can talk about this. You try to make your sound system more audible to everybody.